Bio-Hermes is a study sponsored by the Global Alzheimer's Platform. So the primary objective was to combine or to look at blood-based biomarkers and digital biomarkers to help understand their accuracy in detecting amyloid pathology. So as you may know, brain imaging or brain PET and then maybe a CSF analysis is the gold standard way of looking at amyloid pathology. But obviously those are quite invasive and quite expensive as well, and not accessible necessarily in the healthcare market and so on, which is why there's an urgent need or desire to develop those blood-based biomarkers. And so Bio-Hermes has focused on validating a number of different blood-based biomarkers against amyloid PET together with digital tools. The secondary objective of the study was to specifically try and recruit a high proportion of traditionally underrepresented patient populations. When it comes to clinical trials, what we have seen and what is a very hot topic of discussion at this conference and the previous conferences and in the whole industry is that we see significantly lower proportion of Black populations or Hispanic subjects in clinical trials, and the regulators, the FDA in particular, asking questions in terms of the validity of those studies to approve drugs for the wider population, obviously. So that's an important topic. And again, Bio-Hermes has set a target of recruiting at least 20% of those traditionally underrepresented populations. Actually, that was outperformed with 24% in the actual study. So out of 1,000, roughly 1,000 participants, around 230 were from those populations. And that then obviously links to the presentation that I gave yesterday here at CTAD in terms of the PET findings in that population in particular when compared to the non-Hispanic or Latino white population, which predominantly is recruited in some of those recent AD trials. What we've looked at is amyloid status. So basically whether someone is amyloid positive or not. And then also which is performed by radiologists. Again, that's something IXICO has done as part of the study, where we've collected the data from the sites and then worked with our central radiologist team to have it interpreted for amyloid positivity. And then the second aspect is a quantitative analysis of what is called standardized uptake value ratio, which is basically a quantitative assessment of amyloid uptake in the brain. In this presentation, we've looked at both and looked at those measurements across the different populations. And the key finding that we had here was significantly lower proportion of amyloid positivity in the Black population compared to the white population, which again is something that has been shown by others or has been seen as in other studies. Even in the symposium yesterday, there was one other presentation on the IDEA study where similar observations were reported. And so what we've seen in our study was that we have that difference in amyloid positivity. So the binarist assessment on whether someone is positive or not on amyloid, but not on the quantitative measurement. So we still see on average the same level of amyloid uptake, which of course is interesting because the two are linked and should be linked. And the only way this can be explained is that there are differences in terms of the local uptake. So because if we look at the quantitative measurement, that basically just takes one number across the brain, one average value, whereas when the radiologist makes an assessment on whether someone is positive or negative, they look at patterns of distribution and localized aspects, which probably explain part of this finding. So that's something we'll be looking at in future studies. I guess, what does this mean for the field, or what I should really say is, what new questions can we articulate from those findings? Because I think the big overarching question is, are these because do we see these differences because of differences in how those biomarkers perform in different genetically different populations? Let's put it that way, right? Or are we actually observing some kind of biased and confounding effects because we're recruiting different populations? And so, for example, what has been reported widely is that, or in different studies, is that in Black populations, we see a higher risk of cerebrovascular disease, for example, which again isn't necessarily linked to the genetic differences directly, but could be linked to confounding effects based on socioeconomic status, for example, where again, we know that we have, for example, what was shown in the other talk yesterday was in the Black populations in their study, they had lower household income on average compared to the white population, which is one indicator. What we've seen in our study is that we have slightly lower levels of education in the Black population compared to the white population. And all of these are potential biases that then lead to can potentially lead to those differences. Because if let's say we assume there's a higher this level of comorbidities in the Black population, which could be linked to the risk factors that I've mentioned, then this may explain the level of dementia we see in those subjects compared to the white subjects. And therefore, the fact that they're not actually amyloid positive because they don't have Alzheimer's, but a different type of dementia, if that makes sense.